Welcome, viewers, to what we know will be a fascinating and enlightening discussion from Scientific American Custom Media in collaboration with Albert Einstein College of Medicine. I am Jeremy Abbott, VP and publisher of Scientific American, and we are thrilled to be joined today for a panel discussion with some renowned medical experts. I know that from where I sit in the world of science media, this pandemic has been the biggest event in the careers of science journalists. And I know that for medical professionals on the front lines of critical care and disease research, the same is true. So we are about to embark upon an amazing discussion around the lessons learned and what the medical ecosystem future may look like because of this global pandemic, which into which we are a year. And to start off this discussion, I would like to introduce Dean Tomaselli, Dr. Gordon Tomaselli, Dean of Albert Einstein College of Medicine and Executive VP and Chief Academic Officer of Montefiore Medicine. Dean Tomaselli, I know that you came of age during the HIV/AIDS crisis and had a front row seat to that pandemic. Would love to hear your perspective on world events now and how it can shape our understanding of what we're going through and the future.
Jeremy, thank you. I'm Gordon Tomaselli. I'm the Marilyn and Stanley M. Katz Dean of the Albert Einstein College of Medicine. And yes, indeed, I did have a front row seat to the HIV epidemic as a medical student here at Einstein with my classmate, Lizan Porosky. And then as a resident at the University of California, San Francisco. So did get to see something like this, distinct but like this in my past. We want to thank everybody for joining us in this In Conversation With series. We're so pleased to be partnering with Scientific American in this effort. We began this In Conversation series in late March as a way to provide an update for what was going on in the research space and clinical space at Montefiore Medicine and particularly at the Albert Einstein College of Medicine. It's really been an extraordinary experience for the health system and for the people at Montefiore and Einstein. This pandemic surged with ferocity in March to May. The health system really cared for over 80,000 patients. 12,000 were admitted, 10,000 were discharged to home, but unfortunately 2,000 died. This was met with a truly heroic response by everybody in our health system to gear up, to increase the number of beds, and to provide telehealth services to people who are most vulnerable in our population. We had to change the curriculum at the medical school. Everything went virtual. We graduated part of our senior class early to join the healthcare workforce. Those who didn't volunteered over 10,000 hours of service to do everything from manning PCR machines to doing telehealth for occupational health services, and performing remote infectious disease consults, as well as taking care of vulnerable people remotely. Students also manned 3D printers to create PPE and did all forms of service for frontline workers, including providing babysitting services, pet sitting services, and food runs. We also had to change the way we ran our laboratories. We had to scale back the labs, but we knew that there were a core of laboratories that were either working on viral diseases and pivoted to studying COVID-19, and we established these collaboratories, groups of labs that were involved in either understanding the pathogenesis of this disease, developing therapeutics, or understanding the clinical disease and epidemiology. And this was to great benefit. We developed a unique serologic test, IgA-based serologic test that was approved by the state of New York. Bifunctional antibodies that have high therapeutic potential. Galdesigvir is a drug that was repurposed, designed for another purpose, but is used now in clinical trials outside of the United States. We've developed platforms that were surrogate platforms to SARS-CoV-2 that were safer to use under general circumstances that allowed for studying the mechanism of the virus. We were leaders in clinical trials of antivirals like remdesivir, of anti-inflammatory drugs, and convalescent plasma. And Einstein partnered with the New York City Department of Health to generate upwards of 70,000 aliquots of viral transport medium every week so that broad-based testing could be done in New York City. This was truly a remarkable and inspiring response by Montefiore and Einstein. And we're here tonight to talk to some of the people involved in this effort. This is a very special event, and 3 of our past panelists are reunited for an in-conversation with, 1 year into the pandemic, where do we go from here? Our panel, Jeremy, thank you for moderating it, and I welcome everybody who's joining tonight.
Thank you, Dean Tomaselli. It is a pleasure, and I'm just going to go quickly introduce our esteemed panel and run through a couple of housekeeping notes. Then we're going to have what I think is going to be an enlightening discussion. First off, we have Dr. Lisanne Perovsky, Jacques and Selma Matrani Chair in Biomedical Research at Albert Einstein College of Medicine in Montefiore Medical Center. We have Dr. Michelle Gong, Chief of Critical Care Medicine and Pulmonary Medicine and Director of Critical Care Research at Montefiore Medical Center. Also Professor of Medicine, Epidemiology, and Population Health at Albert Einstein College of Medicine. We are also joined by Dr. Kartik Chandran, Harold and Muriel Block Faculty Scholar in Virology at Albert Einstein College of Medicine. It is a true pleasure to have this panel join us today. For our viewing public, please, you can ask questions in the chat bar during our discussion, and we will have a Q&A at about 30 minutes into this discussion. I would love to start with Lisanne. You know, some people have talked about in the past few years this being the golden era of the immune system. We've learned so much in the past 15, 20 years about the immune system. We now have the general public interested in things like convalescent plasma and herd immunity. These are not things that people may have. This was not the popular lexicon a few years ago. This crisis has certainly increased awareness of so many aspects of immunology. From where you sit doing cutting edge research, how has that affected your relationship in the world of medicine with society at large?
Well, thank you for that question, which is extremely important, and I think gets at the fact that people are becoming interested in science. People want answers about this illness, and they want to understand how it's happening. And I will harken back a little bit to your introduction of Dean Tomaselli about the AIDS epidemic or pandemic, actually, which was another point in time where the public, not as willingly perhaps as has occurred now, ultimately became much more informed about the pathogenesis of this illness. And with that information came a reduction in fear and an understanding that that public health threat could be managed in a way that did not threaten other people, which at the time was a very profound occurrence. And I think many of us who worked on the front lines then were absolutely devastated by what we saw happen to people with a disease that nobody had any idea what was causing initially. And just for a point of reference, it took, depending upon when you date the beginning of the AIDS pandemic, 5 years to find the virus, and about that length of time to develop testing. And one of the things that is incredibly inspiring about what happened now is that the virus was identified almost immediately. And with the identification of the virus came testing. And with testing came an opening of a whole new immunologic landscape that focused on inflammation. And I would say that one of the things that the public has learned about, thanks to the incredible coverage of this pandemic, is the importance of inflammation in the pathogenesis of infectious diseases, which is something that I think is helping people understand why it hasn't been so simple to find therapies. And for me, as a scientist who's worked on antibodies since the beginning of my career, and finally has had an opportunity to apply that knowledge to people, I would say that the interplay between when it is in a disease that the pathogen or the virus is causing illness versus when it is in the disease that the host or the patient's inflammatory response is causing illness, to me, this has been one of the things that has really emerged from this. And I think that that the media has addressed this in a way that it's becoming part of people's understanding of how infectious diseases occur.
A quick follow-up to that. Thank you. So much of the speed that is taking place when it comes to, the wherewithal of the medical ecosystem to solve this, we're building on things we've learned in the past. Because of the HIV/AIDS, we learned a lot about viruses. We are now dealing with a novel virus that affects, the lungs, the heart. We're not exactly sure just what this long-term damage is going to look like. If you look at the cases of pulmonary fibrosis that seem to be happening in certain patients, that's a viral, it's an immune-mediated mechanism that seems to be implicated. There happens to have been a few drugs recently for pulmonary fibrosis. I mean, have we lucked out in some ways because we have made some great progress, from the immune system perspective, viral knowledge? Have we lucked out? Because I feel we've lucked out by having these, we can have a virtual chat. We lucked out by having the internet now, right? So
I think we need a new word for that kind of luck, but I do agree with you. And what I would say is that many of the discoveries that have been leveraged to take better care of patients and to develop therapeutics have been decades in the making. And to me, this is the argument for science. This is the argument for observational science, actually, and for figuring things out and recognizing that there will be a moment when they're important. And two extremely important findings, I think, can make that point. One is that one of the vaccine platforms that is being tested right now is a platform that was developed for another virus. And so that technology, which at the time, many people may have wondered, what are you doing this for? Like, that isn't a very big problem for us right now, is probably emerging as a really great candidate, which gets at the point that infectious diseases are global. This pandemic, I think, is also helping people recognize that, we cannot close off our borders. We cannot keep diseases out. We need to be prepared, and we need to go onto our shelf and find things that may be able to be helpful. And so remdesivir is another example of an agent that was repurposed. So perhaps we lucked out. And as a devotee of antibody-based therapies, I will tell you that all of the knowledge that has been accumulated over the last 100 years is helping us zero in on using this type of therapy. So I suppose we're lucky, but my mother had an expression that I don't exactly remember because it was in Yiddish, but it basically said that luck, was there for the prepared, that it was a combination of knowledge and preparation and being ready. So if that's luck, I would say yes.
That is a great nugget of wisdom. I want to take up that point you made about the importance of global science. And I want to use an analogy from another field. If you don't mind, I'd like to actually ask Michelle this next question. From your perspective as Chief of Critical Care Medicine, after the 2008 financial crisis, and I talk to my friends in the world of banking and finance, financial institutions have to do every month a stress test to make sure that something like that doesn't happen again. And it's a pretty big deal. Not to say that we've solved all financial problems, but has the medical ecosystem done enough of that? And will this moment in our this moment in our globe globally change the way we kind of stress test medical systems?
So I think actually I'm going to address that question on two bases. One, from the issue of the hospital operation issues and the academic missions within academic medical centers, and one in terms of actually the public health aspect of this stress. Okay, so in terms of actually the hospital operation system, this is a unprecedented stress on a system. And the stress was not necessarily even. So like even right now, as we hear about across the nation, many parts of the country, they're being stressed by the number of patients in terms of COVID. Also understand actually that if you look across the map during this year of this global pandemic, you'll see actually areas of the United States and in Europe where they had 3 times the number of patients in COVID that was seen in some other areas of the country now, okay, because it was a bolus. And as such, actually, that's an unprecedented stress that no medical system have necessarily prepared for. All hospital systems have a disaster planning for hurricanes, for pandemics, but there's nothing like a real pandemic to really stress the system. Now, having said that, though, we also have shown the ability of the modern medical system to be able to adapt and to be able to respond as much as possible. And that's a truly heroic effort from all entities across the board. But there's no doubt, however, that stress on the system probably meant that patients who present at the very height of the pandemic may not have as good an outcome as later on when the system is less stressed. And how we deal with that for future pandemics and for future disasters, we're still learning on how to prepare for that in the future. Academically, okay, we need to figure out how in these stress situations is to develop the clinical trials and to do the clinical trials so that we can get to an answer about how to improve outcomes as quickly as possible. We do not have the luxury of saying, let's take care of the medical problems now and the stress now, and then do the trials later. That's not how pandemic works. And that's one thing that this pandemic has been revolutionary compared to actually all prior disasters in the past. I've been part of disaster preparedness for research for a decade and more now. And there's always been issues in terms of regulations, in terms of funding, in terms of being able to just organize, to be able to do large clinical trials and research during pandemic stresses. This pandemic has redefined that landscape. If you just look at some of these global trials like recovery, like what the United States is doing in terms of ACTS and active trials and these platform trials, that is a fundamental departure than how we have been doing clinical trials before. And we have demonstrated that we have the capacity to do that and to launch that in the midst of a pandemic. Yeah. Now, the public health issue is a separate one. And there, we could do better. In many ways, actually, the CDC, the WHO has been preparing for this kind of a pandemic for decades. Yeah. But in spite of that, I think we see from a public health point of view that there are still elements of collaboration, of communication, of organization, of mobilization that could be improved and worked upon. And there, okay, we still need to work on some of that preparedness.
Yeah, I can imagine, again, refining what we define as preparedness will change. I want to take this over to Kartik for a moment, and maybe even talk about your research at Einstein and how you've had to pivot. You know, I know that you worked on Ebola, and you know, when this pandemic hit, how were you able to quickly kind of shift gears? How has it affected your research? And you know, in light of what we were just talking about, you know, how will your research hopefully impact how we deal with things like this, hopefully never this bad, in the future?
Yeah, thanks, Jeremy. So I just want to very briefly touch on what Lisanne and Michelle said, and it will sort of connect with what I'm going to say in answer to your specific question. So first of all, we prepare for pandemics in between pandemics. It's sort of like having a fire plan. You can't make it up, you know, once the fire starts. You need to have a plan in place and that you can execute. And so I think that's something that we are going to have to work on at all levels, at a global level, at a national level, at state and regional levels. And you know, my expertise is really, you know, understanding how viruses work and then trying to develop therapeutics against them and a little bit of vaccine work. And in that regard, I think also just to follow up on what both, you know, Lisanne and Michelle said, you know, we've from not just studying HIV, but also Ebola, you know, for which we now have antibody therapeutics, for which we now have a vectored vaccine, and a number of other sort of agents, you know, we've actually, and also we've learned a lot from the cancer field about antibody therapeutics, not just how to find them, to tune them, make them better, but also how to make them at scale so we can give them to people. So hantaviruses, Crimean Congo hemorrhagic fever virus, these are viruses that the WHO cares about because they are, you know, present in many, many different animals. They're vectored sometimes by ticks, sometimes directly transferred from animals to humans. And these are all potential zoonotic threats, meaning that they can start a human outbreak by jumping from an animal host to a human, right? So, and you can put this virus as well as the original SARS, as well as MERS, you know, other coronaviruses in the same category. So I think we'd already been thinking about a couple of different aspects of how you sort of take what you know about a new virus and use that information to understand what makes it tick and how to make a drug against it. So the first step is you need a way to study it. And Ebola is worse than COVID because it's a BSL-4, it's a biosafety level 4 virus. So you really need these high-end facilities. There's not that many. It's very slow to work, as you can imagine, because you're in there wearing spacesuits. So, you know, it's a bottleneck, you know, to actually do anything there and do it quickly. You know, COVID's a little bit better because it's BSL-3, but the same sort of principle applies, which is that, you know, it's just much, you know, it takes a while to gear up and, you know, really do these things quickly. So we need a system that we can work with in a regular lab, what we would call biosafety level 2. And that's where kind of, you know, one of our expertise comes in. We had built such systems. Essentially what we do is we build what we call a sheep and wolf's clothing. So we essentially take an innocuous virus, strip off its outer shell and replace it with that of whatever virus we want to study. And so we did that for Ebola. We've done that for hantaviruses, a bunch of other viruses. And so, you know, we very quickly moved, you know, once COVID started. I mean, essentially, we had a conversation in my lab at the end of February about what to do. We were supposed to go to a conference in San Diego, and I basically made a unilateral call that we weren't going to go to that conference because, you know, I knew this was going to be bad. Actually, you know, my wife knew this was going to be bad even before me because she worked at a hospital for a while, and she was like, you know, you're a virologist, but you're not really thinking about the public health aspect of this, that this is going to be bad. There are still people out there, you know, eating dinner at crowded restaurants and going to nightclubs, pretending like nothing's happening, but you know this is going to hit. So prepare our family and prepare your lab. And that's what I did. And, you know, one of the things we did was we sent about half the people home, and then I asked for volunteers. And so we had a group of 8 people that we call the COVID crew that decided they would come in and keep working, wearing masks, you know, and really build some of these tools. So the first thing was we built this surrogate system to study the virus, which now turns out to be extremely useful for screening drugs and antibodies, all kinds of other therapeutics, even as a vaccine vector. And then the second piece of it was that, you know, Lisanne and I know each other. We've known each other. Lisanne, I remember meeting with Lisanne when I was interviewing for a job at Einstein as an assistant professor back, you know, 2006. And, we haven't really worked together, but Lizanne and I connected because Lizanne told me, we need serology. We need to be able to look for antibodies in convalescent donors. So we very quickly sort of retooled the lab to try and develop a test. And again, it's this intrepid group of students and postdocs in the lab who came in every day, even though everybody was sitting at home. And it was a little bit like I Am Legend out there with like nobody on the streets. And these people came in, every day and worked like 16 hours a day to actually get this assay built that we were then able to very rapidly, in a larger collaborative effort that included people in Lizanne's lab, Jonathan Lai's lab, really rapidly screen all of the sera that were coming from these potential donors, which allowed us then to prioritize people to give plasma for convalescent plasma. And from that, we actually work with people in pathology, Mike Prostowski, Amy Fox, Lou Weiss, to actually develop an antibody test that could actually be run at our hospital. So, and then we are involved in a number of other sort of activities.
We keep hearing about turning the corner. And I want to, would your research bear out that we are turning a corner? You're working in a very provocative area. Convalescent plasma has some mixed results, but there's a lot of good news. And of course, there's also news coming in a lot about vaccines. So where are we in this corner from your perspective, Lizanne?
Wow. Well, that's a multifaceted question, but let me just back it up a second and pick up on a couple of the comments that Kartik just made. And this also is a shout out to our dean. We were able to really be major players in thinking particularly about antibody-based therapeutics because we were able to still come to work, because we had resources to develop platforms to diagnose or to look for antibodies, as Kartik mentioned. And because honestly of the enormous spirit of collaboration that has been very, very long-standing at this place. I too am a graduate of Albert Einstein College of Medicine. But what I would say really propelled us to think about these things in an innovative way is how we brought the clinical operations and the research operations together. Johanna Daly, who is a colleague of ours, developed an absolutely inspirational donor program, which really was built on the courage of the community that was probably the hardest hit and that suffered tremendously, both from disease and because of perception early on in the pandemic about their illness. And these people came forward and they donated their blood, and we were able to use Kartik's platform, which is just a highly specific, amazing assay, to be able to look for antibodies. And so our convalescent plasma program was born. And we were able to, from the very beginning, no matter what sort of a study we used, to deploy very, very high titer plasma that we were able to identify in that way. So the studies that have been done have certainly shown that it's safe. And, Kartik mentioned safety, and that's really, really important. The second thing that the observational studies of convalescent plasma have shown is that they've reinvented 1918. They've shown us that using it in desperately ill patients who are intubated and whose prognosis is very poor, it is not going to be the right window for using this stuff. And so I would argue that we've learned an enormous amount. We personally, I with a number of colleagues across New York City and now across the country, with the support of the NIH and Operation Warp Speed, are conducting a randomized control trial where we are comparing convalescent plasma to a placebo, which is saline in hospitalized patients. And the way that the story has unfolded, I think that we are all very, very anxious to know where the window for using this is going to be and how effective it may be in hospitalized patients. In terms of my own efforts, for me, maybe this is a bit of that kind of luck that we need a new word for. I've worked on antibodies for over 30 years. I walked into Maddie Scharf's lab, shell-shocked from the AIDS pandemic, having never touched a pipette, wanting to become a scientist. And I focused on antibodies because he let me in his lab, thank goodness. And I learned about antibodies, and I've worked with antibodies in mice for all of this time. And this was my opportunity to try and apply a lot of the things that I had learned and felt were important to humans. And I'm really proud of that. I'm happy that I've lived long enough to be able to do that. The other thing that I have to say, and it really has to do with the community of science and what an enormous inspiring enterprise it is to be involved in, is that my laboratory was basically converted, as Kartik's was, into a serology laboratory. And basically all we've been doing for the past, however, since March, has been to titer plasma and to get a better understanding of how the antibody response is occurring in people. And as this has been going on, we went from people thinking antibodies did nothing to a point now where, both from data that has emerged from monoclonal antibodies in other areas, and even in hospitalized patients, we see that the endogenous antibody response is playing some sort of a role in clearing virus early on. The cart and the horse get separated in people that can't control it well enough. But to me, this is one of the most resounding endorsements of a very long-standing principle that we really haven't been able to prove, that the initial antibody response to a viral infection is extremely important in controlling that virus.
That's a big lesson for science. That's a big lesson for science.
Absolutely. And I mean, if I may, there's just one other thing that I want to say, which is that science is really expensive. And we've been in an era where, both for clinical trials, as Michelle referred to, as well as for bench science, we've faced a real crisis in terms of the amount of funds that are available. And I really do want to shout out to our dean and donors at Montefiore, various foundations that really funded us early on, kind of on a wing and a prayer. None of us had really done this before. None of us really had credentials. I mean, Michelle has credentials as a clinical researcher, but what do I know about viruses? And I think that we'll forever be grateful for that investment. And ultimately, the federal funding did come along. I hope it wasn't too late.
Great. I'm going to pick up your point about safety and throw it back to Michelle for just a quick discussion about public perception and safety. And of course, the big question is, when a vaccine is proven to be effective and safe enough, what is the challenge in convincing the public that it's safe?
So I think that's a very relevant question and very pointed. And that's partly actually because we may be at a stage in modern era where there is a greater doubt of science, just as we're actually are relying more on science to be able to come up with the solutions. And I think that's a challenge for all of us, right? Us as clinicians who take care of the patients at the bedside, us as clinical researchers, as well as actually, Kartik and Lisanne being basic science researchers and translational researchers, and you as the editor of a major scientific journal, and how we communicate. Because we need to actually communicate beyond just our scientific community now. And we have to be able to translate what it meant when something is approved because of a clinical trial. So some of those reservations about vaccine, which predates actually this pandemic, comes from a lack of understanding or about the difference between an anecdotal observation versus a clinical trial, where you're actually comparing patients and outcomes. And anecdotal is much more powerful on social media. The story is a lot more powerful It resonates a lot more than if I say, well, the relative risk is 1.4, and with a confidence interval that falls well within significance. Because of that, we have actually maybe have not taken as strong a role in understanding how to communicate the scientific findings about risks and benefits to the general lay community, forcing many of them actually to look online for whoever is maybe the loudest or sounds the most strong in terms of their proclamation.
Yeah.
And that's something we need to work on.
Yeah, that's a great answer. I'm gonna answer my own question, and I'm gonna throw a question to each of you, because Michelle, you've crystallized what I'd like to be the biggest lesson from the world of science media and the public understanding of science, which is to appreciate the process of science, to understand what clinical data means and what clinical how to process that. And I'll start with Kartik, because you've been silent for a few minutes. If you could boil down in maybe a minute or so, maybe a minute or less, what you'd love to be, what kind of the biggest impact, what we can learn from this and how it can help to affect change in the medical ecosystem in the future.
Yeah, for me, there's, as somebody that works on emerging viruses, the real lesson here is how do we get ready for the next pandemic, or maybe even the one after that? And so from a sort of a therapeutic and vaccine side, what structures can we build that will essentially have us be ready the next time, so that we have something on day one instead of having to wait till day 90 or day 120. And so to me, that's science, it's platforms, research platforms, it's working with government and industry to have structures in place to stockpile things, manufacturing, and then it's all of the other pieces that you need, the federal, state, and local, like the international collaborations, the sort of the interactions and the coordination, to actually to develop, test, and deploy. I don't know what's a reasonable within 60 days. I mean, to me that would be, or even sooner, maybe 30 days, right? So there's tons of other coronaviruses out there in bats. I mean, there's tons of viruses out there. In the last 20 years, we've had 6 different coronavirus outbreaks. People only think about the 3 in humans, but there were 3 in pigs as well. And all those pig viruses can infect human cells. So there's a lot more of these where they came from, and that's just coronaviruses. There's all kinds of other things out there. So we need to be, we need to invest at all levels, including educating, people, the public, and that starts with K through 12 on, and really to be ready for this, because this is going to be a fact of our lives going forward, just like forest fires in California or climate change. In fact, they're all connected.
That's a great lesson. Lisanne, I'm going to throw it back to you. The biggest takeaway you hope from this experience, and how it can hopefully the impact on the medical ecosystem of the future.
Wow. I think I have to pick up on what Michelle just said. My biggest takeaway is that knowledge and understanding are the best way to combat fear and to protect ourselves, and in this instance, make our society safe. And for all the talk of science and everything, I really am struck by the importance of basic public health principles and how important they are in controlling this virus. And so for me, the takeaway is to find better ways to communicate that message, which will, if you'll remember early on, we talked about flattening the curve so that we had time to do whatever. And I think that we're in a moment where we need to remember that. And so what I would say is that I hope that this is another word that we probably need a better one for, the rebirth of basic public health principles, which by the way, go back to the plague in the 14th century, to find better ways of having the public understand the importance of these principles and the enormous good that they can do. And I think that the final comment on that is that, you know, this, for various reasons, has made me a believer in the goodness of people. And I'm really hoping that those two things can come together. And for me, that's the take-home message.
That's a wonderful sentiment. And Michelle, you want to close out on what your lesson is before we go to the Q&A?
Yes, and actually, I want to take off on what actually Kartik and Lisanne just said just now, and which is actually, look, we all have the same goals to improve the health and decrease harm among all of our patients. But what we have recognized from this pandemic is that there is a real need to turn our modern healthcare system into a learning healthcare system where we can integrate the basic science, the translational science, the clinical trials into our clinical operations, both so that patients can get an understanding of how research influences their care and what type of care they should be getting, but also actually because that's the fastest way for us to find answers to how to improve outcomes in our patients, and the fastest way to translate the scientific findings into practice, which is ultimately all of our goals.
That is a great lesson as well. And we're gonna pause here. We're gonna get to a Q&A. This was an amazing panel discussion, and I'm happy to kind of bring in the viewers. I also would like to see if Dean Tomaselli wanted to make any final comments as he's been a silent observer of this before we go to the Q&A.
Not only silent, but proud. And I want to thank all the panelists for presenting today. It was really, truly amazing work. And I think whether or not we're talking about Hindi or Gujarati or Cantonese or Mandarin or Yiddish or Italian, I think what we're looking for is chance favoring the prepared mind. And the prepared mind is investment in biomedical research. That's how we are prepared. That's how we can turn on a dime. That's how we can figure out and determine the DNA blueprint of a virus like SARS-CoV-2 in 24 hours after its isolation. It also makes me wonder why we're not investing more than 1/4 of 1 cent on the dollar in biomedical research based on our GDP. So I think that the lessons for me are that biomedical research has paid us back many-fold over. And I think we ought to be thinking about investing more in science in general, but in biomedical research in particular, if we really want to be ready for the next series of pandemics that we're afflicted with. So again, thank you for allowing me to make a comment. And let me thank our panelists and you for really an inspiring discussion.
Thanks for those great words. We're going to go to a Q&A, and we've got some great questions that are flooding the inbox already. So I want to get to the first one. And maybe this is a question for Michelle. There was a surge, obviously, from March to May in the New York region. Other surges happened, a little bit after that in the U.S. We are now faced with a potential surge upon a surge with Thanksgiving travel, and perhaps even a surge upon that with more holiday travel potentially in store in the next few weeks. Are we in danger of testing the capacities of our medical facilities? The crisis that we assumed might happen earlier in the year didn't come to pass exactly, though it was pretty scary. What does it look like for the next couple of months?
So that's a great question. I think that overall we're in a much better shape than we were back in March for a number of different reasons. For one thing, the scale of escalation in terms of the number of patients we're seeing is not quite so steep as it was back in March. And most of the deaths and most of the patients that you heard about back in March were concentrated in small areas like New York City and certain parts of New Orleans and other places. Now they're kind of more spread out throughout the country, but the rest of the country has also had a bit more time to prepare, to get the resources that they need to be able to deliver the care that is needed by these patients. In addition, since that time, we've had a number of successful clinical trials that has added to the treatment armamentarium for COVID. So we're now going into the second, third wave, if you will, with more treatment, better understanding how to manage these patients, and to better understand how to prevent complications. So even as we're seeing more patients with COVID, we are seeing less of them being very severely ill or having multiple organ failure. And that's a good sign of us learning from our clinical trials and our clinical research from even the early days of the pandemic.
That's a great point. I'm going to throw this question, which comes to us from Ronald, over to Kartik, and if anyone else wants to weigh in, why don't we have a daily do-it-yourself litmus test type of, distributed to all test? There's a lot of confusion on testing, and where is it going to be, say, in the next couple of months?
Yeah, that's a really good question. I agree. With Ronald, I really think, it would be really wonderful to have such a daily kind of disposable test that you could run. I think there are several such tests that are in the works, but I think, there are a number of different issues. I think there's some confusion about how you actually deploy them effectively. One problem with these tests is that, although they can be very specific, which means that they don't tend to pick up other things, they're also not as sensitive as sort of the gold standard tests that are being used, the RNA-based tests that clinical labs are using. And so, there's a concern that, positive, that people are going to show up false negative. But I think that's sort of allayed by the fact that if one gets tested every day, like if you test yourself every day, you're very unlikely to get 4 positive tests in a row. So that kind of gives you some confidence, even though you can't necessarily rely on any one given test results. So I think that there is a paradigm that would allow this to work and to actually to detect the virus at home. But I feel like right now we're like in the uncanny valley a little bit, where these tests need to be really cheap and really available and be able to be manufactured in huge numbers. And we don't really have that right now. We have tests that are really good and really specific and sensitive, but the supply is not there. And then we have other kinds of tests that aren't as sensitive. So you can't really use them in the same way you would use like a qPCR standard test. And the way you get around that is by doing repeat testing over and over again. And we simply don't have the distribution and the manufacturing to really make such a test available. So we're sort of in between, and I think that's changing in the next few months. Hopefully we're going to get the kind of test where you can, just test yourself every day at home, because that would make a huge difference.
Yeah, as you guys could, great point. Look forward to as that evolving. Lizan, I'm gonna throw this one to you, and when I say this one, we've gotten several, as you can imagine, about vaccines, and some of our viewers want to know your opinion about which vaccine is the most reliable, and what's in store for us vaccine-wise with all this news coming in.
I will echo my colleagues by saying that's a great question. And there are a number of very promising candidates, vaccine candidates, some of which are on different platforms. I think that we are on the verge of being able to deploy the RNA-based vaccines here in the United States. And I really consider this to be nothing short of a miracle. The efficacy of these vaccines is astounding. Their safety appears to be acceptable. We'll learn a lot more in a few days after the emergency use authorization is evaluated. I think we will ultimately have a number of different vaccines that may be on different platforms. And I think as many who are listening have heard, that's a good thing. Some of them may be better for certain types of recipients than others. And as the data are accumulated, we will be able to use some evidence-based, some evidence to be able to make decisions about this best vaccine. I will just harken back to an earlier part of our discussion about vaccines and about these RNA platforms, and really say that as somebody who has been interested and worked on vaccines and vaccine responses for a long time, this platform is so exciting. And it's been a long time in the making, although it may seem like, this just came so quickly. These types of vaccines have been used in investigational studies for a long time. They've been studied intently in terms of their how they work. But the actual carrying through of this platform was really due to the dedication and commitment and belief of the scientists who worked on it. So I think that that's a very important take-home message about this particular platform. And we'll have to wait and see. We'll know a lot more in a couple of days, but I'm very, very optimistic that the vaccines that we have now that will be undergoing emergency use authorization soon will really be an incredible step in the direction of preventing disease and possibly preventing.
Well, that's great. The world awaits, and we certainly are many eyes looking to the UK today and in the next week or so. Real quick reminder, because there's a little noise on the system, if you're not speaking, if you guys can try and mute, that would be awesome. But I want to involve everyone, so of course there'll be a lot of muting and unmuting. Here's a question. It's come in different ways. I'm gonna throw this question over to Dean Tomaselli, over to Gordon. This is a question from Dan. What could the WHO and the CDC do better to improve pandemic responses next time? As someone who's been through a few pandemics, would love to hear your perspective on that.
That is a good question as well. I think we can't rely on mitigation, that is for pandemics and viral infections to be widespread before we start to do something about it. We need to be surveilling and on the ground and preventing things from, as Kartik mentioned, zoonotic infection and jumping from animal reservoirs to humans is going to be a fact of life. We need to recognize that that's a fact of life. We need to be globally surveilling for things, contain them when they're first detected, rather than trying to mitigate once they start to spread. So I think this is not just a United States effort, it's a global effort. It requires global partnership, and it really does require being vigilant literally around the world. We were on the ground, or should have been on the ground in Wuhan, to really keep this virus in China, but it turns out that we weren't this time. But I think that's a lesson learned, and I think some of the prior pandemics we did a bit better job at controlling things in place so that they didn't spread as widely. But I think kind of a global effort of surveillance is really what we need to be doing, not only institutions in the United States, but worldwide.
Great point. This I will bring back over to Lizanne, but others can weigh in. We have been awed by the speed, as we noted earlier, at which these vaccines have come out. Can other areas of healthcare and medicine take a lesson from some of this? I mean, this question came up a couple of times, but it's also one I've had. You've got the government giving a big pot of money to different organizations, institutions, companies, saying, we're going to give you this even if the vaccine doesn't work. That's a pretty nice incentive. Obviously, that's not always going to be the case, but are there other collaborative elements that areas of health could take a lesson and maybe speed up the development for other things in medicine?
Absolutely. And I think that this, the rollout of the vaccine from the early animal studies, the phase 1 to the phase 2 to the phase 3 trial, showed us that if we do things semi in parallel in a situation like this, and we're willing to take the risk of something not working or not leading to the expected outcome, we can do this. And so that is one element that can enable very rapid development of therapeutics or other interventions. But there's another thing that's very important, which is the funding that Dean Tomaselli mentioned. One of the reasons that this process has been able to occur is because of the investment that was made in it. And then the final piece that enables very rapid discovery and development is leadership and collaboration. And I would say, hearkening back to some of the comments that Michelle made earlier, and also to some degree some of the comments about testing, is that I think one thing we need to take from this is that we really do all need to work together. That this is not a question of individual credit, and that a sort of cottage industry investigation here and there is not well suited to a pandemic. And so very broad-based collaboration, discussion, and coming up with teams that can work quickly to implement things with adequate support is a model that I think can really go forth. And not only in therapeutics and in care, but honestly in everything that we do, how we train physicians, how we bring people into medical education. And we've been sort of taking stabs at team science for a very long time, but I think that this has really convinced us about how important it is and how successful it can be, and how the timeline can be collapsed, not because anybody's cutting corners, but because there's an enormous amount of efficiency that accompanies processes when people work together.
Yeah, that's a great point.
I just want to add one thing.
Please.
To that. I mean, I think it's important to consider the difference between what you do during an emergency and what you do in a regular situation, right? So, what we're seeing right now is this highly decentralized sort of, Manhattan Project for COVID that's happening because, we're in the middle of a global crisis. And, this is one of those few times in recent human history where every single person can relate to this, right? But when things go back to quote unquote normal, then, everything's going to get balkanized again because different people are concerned about different things, different governments. So I think we'll have to find a way to navigate that landscape while keeping in mind the lessons from COVID, while also, I think, recognizing that, we can't do this for everything. So, we could, I suppose, if we had enough funding. So Lizanne pointed that out and Dean Tomaselli pointed it out. So, but, what do we do once, kind of the world's eyes are not all focused on one thing, and how do we learn from this and leverage what we've gained here? I echo Lizanne's, what Lizanne said about team science, because I think that is definitely the way forward.
Well, just one other very quick thing on that, because Kartik raises a very interesting point, and we all know that one size doesn't fit all. And so, disparate efforts, lots of people thinking about something differently, the diversity that comes from that is really, really important for coming up with ideas, coming up with a path forward. But the getting across the finish line, the getting to the next stage of proof or discovery, it involves a different kind of diversity, and there it really involves a lot of people working together, having very defined roles, and really working towards the greater good.
Yeah. We've got time for a couple more questions, and they're flooding here. This is a fascinating question. This is from Michael. He's a doctor in the UK. Gordon, if you want to take this one, and certainly if others want to weigh in. Michael writes, as a doctor in the UK, I'm seeing the rollout from today of the RNA vaccine. The biggest question in the minds of the people here is, what are the longer term effects of this type of technology going to be? If they have been used in the past in previous studies for different reasons, what evidence is out there to try and allay some fears? Fresh from the UK, doctor.
Well, thank you, and congratulations, because for the rollout of a vaccine. in the UK today. We're looking forward to doing the same thing here in the United States in the short term. Of course, we can't know definitively what the long-term effects of anything, of any therapeutic is going to be without observation over a period of time. What we do know is that the short-term benefit in preventing illness outweighs the short-term risk, certainly, at least with the candidate viruses vaccines that we have right now. So again, I think there is a very low frequency of side effects so far. Everything looks actually pretty good. And as Lizanne mentioned, this is a new vaccine platform that we're working with now. So I think there are things that we're going to learn over time. But I'm confident, and I feel comfortable personally rolling up my sleeve and getting vaccinated as soon as I possibly can with the vaccine platforms that are out there now. I think the bigger problem for many of us is the doubt and the basically some of the questions that the general population has about getting vaccinated, because we are going to have to develop immunity, whether that's natural or vaccine-based immunity in a large portion of the population to really control the pandemic. So it's going to be important, as somebody mentioned previously, that we are not only scientists, but we are messengers.
Mm-hmm. You know, the vaccine rollout is certainly one piece of the puzzle, a very big piece of the puzzle. Then there, of course, is the supply chain, keeping these cold with the freeze boxes, vaccination sites that have to be set up, scaling, making sure we've got scalable production that's sustained. You know, Michelle, what do you see as the biggest challenge after, you know, the glow of this vaccine, which is amazing? What is the biggest challenge? I mean, are they all just equally going to be challenges in the next few months?
What you point out there is the differences between a scientific discovery and then the implementation of that into care. There are many challenges, as you mentioned. You know, the storage, the freezer space, the delivery. But at the same time, these are operational challenges that are not insurmountable. But it does require a certain amount of leadership, because, you know, even issues such as, say, enough freezers, enough kind of people to be able to deliver these vaccines to the people in need. If there is a unified will and organization willing to put together, you know, people and orders the companies to wrap up production of freezers or what you need, it's all possible. And it is all doable. And in many ways, the United States, the UK, many countries have done something similar in the past. So I'm actually very optimistic. I think one of the things that's also unique about what we're happening now is that collaboration that Lizanne was talking about, it extends not only to the scientists from different fields, but also actually to industry. This collaboration between scientists and industry in testing and development also means actually we can leverage the operational expertise in large-scale production and dissemination using those companies.
Yeah. Well, it's going to be quite an effort, and one that the world will be watching this collaborative effort. Here is a question coming in from Dan, and I guess this is probably most relevant for you, Lizanne. Are there lessons from the research on convalescent plasma that could offer clues to development of monoclonal antibody therapies for COVID-19?
Well, thanks for that question, Dan. I would say yes, although it may actually be the other way around. So what we've learned with convalescent plasma is that it is essentially functioning as an antiviral, and it works best early in disease. It works best before that inflammatory phase of the disease that I mentioned earlier begins to occur. Monoclonal antibodies are antibodies that bind to a very specific determinant And in this case, they bind to the spike protein, or we're focused on monoclonal antibodies that bind to the spike protein. It's interesting that we have learned from the monoclonal antibody studies in the outpatient space that they appear to be effective, and they appear to be effective particularly in people before they have begun to develop their own antibody response. And so the information that we kind of have from both modalities is really helping us zero in on a time of illness when any sort of antibody therapy is likely to be effective. What we may be able to do that is very exciting from convalescent plasma is to be able to isolate antibodies that may bind to different determinants or that may work by mechanisms other than neutralization. And so the hint that that may be going on is that even in studies where convalescent plasma does not appear to, quote, work, where it has not reduced mortality, for example, compared to a control of some sort, it almost universally reduces the viral load and can reduce inflammatory signals of inflammation like cytokines and reduce the CRP and raise lymphocytes. So the way that monoclonal antibodies, that convalescent plasma may inform other monoclonal antibody therapy is the ability to kind of fish out the antibodies that are mediating this particular effect and then raising antibodies that may be, for example, very useful against inflammation, so that we may be able to have on our shelf antibodies that are useful in inflammation, so late in disease, antibodies that are useful against the virus early in disease. There are very exciting efforts going on right now dissecting the different types of antibodies in convalescent plasma using a kind of system serology approach, which has been pioneered by Galit Alter at the Ragan Institute. And we are currently collaborating with her to look at the convalescent plasma that we're giving to patients in our trial to see whether we can pick out antibodies that may be.
Well, we look forward
to add to that.
Oh yeah, please.
So, I mean, just to follow up on what Lizan said, I think there's definitely value in developing monoclonal antibody treatments that aren't doing sort of what the majority of the antibodies in our own immune response are doing, because, I mean, some of the leading candidate monoclonal antibody treatments are targeting certain parts of the spike that are also seemingly targeted, quite extensively by our own immune system. So there is sort of a longer term issue there potentially where, just because viruses evolve and change in response to our own immune responses, if the monoclonal antibodies are sort of doing the same thing, they could be vulnerable. So I think there's something to be said for making antibody treatments that actually sample something very rare that's happening in the blood rather than something that's happening most of the time. And I mean, HIV provides a great example of this, where the therapeutics that are antibody-based for HIV that work are really ones that are very rare to find in people. And you really have to go looking for them. I don't think you can really say that about the first generation of monoclonal antibody therapeutics against COVID. So I think there's a lot of work to do there to sort of find these second and third and fourth sort of iterations of the antibodies that are doing something that's not sort of the majority thing that's going on in the immune response, if that makes sense.
Yeah, that doesn't, again, the world will be watching as these therapeutic modalities unfold, along with everything else that is going on. And no doubt the events of this past year will profoundly affect what is going to happen in the next year, how medicine is taught, how the public perceives science. This has been an amazing discussion. We are out of time. I wish we'd gotten to more questions, but I want to thank everyone involved on this panel, this esteemed panel. Really, thanks for the time and the insight. I want to thank our viewers for the great questions and for logging on tonight. And of course, we want to thank Albert Einstein College of Medicine Women's Division for developing this partnership with Scientific American. I was really honored and pleased to lead this discussion. So thank you everyone for the time, and we will see you again at our next, hopefully we'll do another In Conversation With and other events from Scientific American. This was great, guys. Thank you so much.
Thank you. Thanks, everyone. Good night.
Thank you.
Good night.
Good night.
Good night. Thank you.